students. I'm Alice Preeti, Assistant Professor, and I will be the host for today's program. First of all, a very warm welcome to all of you gathered here for the Industrial Expert Talk on Gene Editing in Drug Discovery and Career Mapping. This has been organized by Padma Shri Institute of Management and Sciences. The theme for today's talk is titled as Gene Editing in Drug Discovery and Career Mapping. Our special guest is an industrial expertise, Dr. Keerthana M.V. Shinde, Commercial Marketing Manager, Customer Experience Lab, Merck Life Sciences. I would like to Now I would like to call upon our principal to talk a few words about our speaker.
A very good morning, and I welcome you all to this lecture series. We thought of giving you exposure for 75 lecture series in this 75 years of marking the thing of 75 years of independence. Maybe we are going ahead with 10th or 12th lecture from the industry and repeated institutions, and also from various walks of life. They are here actually to share their experiences. I wish you will take the advantage of it and you will learn from their experiences to carve your future. With these few words, I welcome you all for this industry expert talk, and I welcome Keerthana for our campus. And she looks quite energetic, young, and has got vast experience in gene editing and discovery, drug discovery, that in companies like corporates like Merck. There will be a lot to learn from such people, to listen from such people. I'm eager to listen from Ms. Keerthana. Keerthana, welcome to our campus, and I welcome you all once again. Thank you.
It gives me immense pleasure to introduce to you our guest, Dr. Keerthana. Dr. Keerthana is a prominent cancer immunologist with over 11 years of experience in drug discovery and drug development. She carries 2 patents, 3 lead novel biologics, and 3 lead small molecules under her name. With the obvious facts that she is a scientific expertise in the field of molecular biology, functional genomics, proteomics, and immunoassays, she has completed various research projects, seminars, workshops, services, and has trained over 500 people. Dr. Keerthana was awarded a PhD at the Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. She further went on to pursue her postdoctoral research at the University of California, San Francisco, USA. She was also the former scientific manager of Zomato Biologics and is currently the commercial marketing manager, customer experience labs of Merck Life Sciences. Her technical skills are innumerable, ranging from techniques in bioassays, molecular biology, protein analytics, bioinformatics, cell biology, to name a few. It gives me so much joy and pride to have you with us, ma'am. Gene editing systems are one of the most powerful tools that have been well established by researchers in search of new drugs. From zinc fingers to CRISPR-Cas9 systems, the technology has been firmly established and scientists are exploiting to find new drugs for diseases. With this, I request our guest of honor, Dr. Keerthana, to take over and deliver a talk. Over to you, ma'am.
Everyone. How is everyone doing? Going to sleep or going to stay awake throughout the talk? Okay. So first of all, I thank Dr. Iku for inviting me. I connected with her last month, and your beloved principal, ma'am. Thank you so much. You have really been in last few minutes of interaction. I could really connect so well with you, and the expertise that you bring to this institute, I'm sure all of them are going to fly very high in their career. So today I'm going to talk on gene editing in drug discovery and career mapping. As your principal ma'am said that I'm young and energetic. It's true what she said today. And in the second half of my talk, I'll tell you why I look young and energetic. So you will all know the reason, the secret of it. So going on to the talk, so as I come from Merck, I'm sure all of you have heard of the company, but I will give a quick overview on it. So currently I'm commercial marketing manager for customer experience labs. I want you all to know what is this customer experience labs. So Merck, we are vibrant science and technology company. We are leader in life sciences. We have vibrant team of specialists in healthcare, life science, and performance materials. We are dedicated to solve the toughest problem in life science by collaborating with global scientific community. So these are the two customer experience lab, what we call as Merck Innovation Lab, which is in Bangalore towards Electronic City. My office is located there, and we have another lab that is Merck High Skill Development Center, which is in collaboration with the CSR MTech in Chandigarh. So these are the two labs, which is Merck's vision. What do these labs do? They want to support, enable, and facilitate life science research in India to provide tools and techniques to the young force so that we can build the skill sets that's needed for their successful career transition. So I currently head these two labs. We have the vision to establish more labs across India. Hopefully that will happen in next few years. But currently, these are the two labs that we have. And what's our purpose? We are building life science skills in the country to enhance scientific competency and advanced technologies. We want to establish Merck now as a thought leader in the field of scientific skill development and collaborative research. So that's our purpose, and different working areas that we have for the lab is molecular biology, cell biology. We have high-end equipments in cell biology, something called Receptor 3.0, which is an automated cell counter. We have cell ASICs, we have millicell DCI for imaging and different, providing external environment for cell growth. So biomarkers, we carry a huge portfolio. As you all know, these days, a lot of therapeutics that or treatment for the patients is decided based on the biomarkers. You do patient stratification, which therapeutic actually works on the patient. It's not based on just the disease now, it's also based on what kind of biomarkers they carry and what kind of therapeutic gets applied to those particular category of patients. So we have a huge portfolio in biomarkers, and these are all the high equipment that we have, such as SMC X-Pro, which does a femto-level detection of a biomarker, which is not actually possible by any other instrument. So this is a special equipment that Merck carries. We have Auto 2D, we have Snap ID, where you can do Western blot within 30 minutes. Anybody here who does Western blotting? Okay, so we have Snap ID where we cut down, I'm sure considering your blocking time, primary and secondary incubation, it would take 1.5 days to complete a Western blotting. Whereas this high-end equipment that we have, we can do it in 30 minutes. So it's basically for research scholars, or it could be a project, a high demand project where you want to actually cut down the time to deliver. You might have deadlines. So these kind of high-end equipments. And definitely we have functional genomics. Today I'm going to touch upon a lot of gene editing techniques, which is very much critical these days in drug discovery. And we have a lot of skill development programs. I'm not going in detail. This is to just to give you an overview what these labs do. We have digital training, certificate workshops. We have hands-on trainings. You see, we have basic and advanced training programs in all these working areas. So you're welcome to come and get trained at our labs based on the topics that will interest you. So far, how the labs have contributed, as I said, we want to solve the toughest problem of our scientists and researchers in India. So we have been collaborating even during the COVID time for vaccine development projects. We've analyzed samples for biomarker analysis and also in genomics and protein research. We have done 5,500 plus sample analysis, and in skill development so far we have delivered 250 plus lectures. We have 12,000 students who have participated in our training programs covering 1,000 plus universities and colleges. We have signed 5 MOUs. We have 17,000 students with us as a database where we keep sharing the upcoming events. And importantly, there is a LinkedIn group. So at the last of my presentation, I'll share the link, whoever wish to join. This is a community of all the academicians, scientists, research scholars, and students. This is a private LinkedIn group by Merck, and we already have 7,500 students there. So and research scholars, you're welcome to join there to get most benefit in the skill development programs or even the job transitions that you want to do. So today's talk, the overview goes like this. We have gene or genome editing techniques. I will mainly focus on CRISPR-Cas9 system. We'll also focus on cancer therapeutics. So if you look into the drug discovery, CRISPR-Cas9 system is used very, very effectively in cancer therapeutics, and it has really transformed drug discovery. So you will understand how it has revolutionized and evolution of cancer immunotherapy. Like I'm sure all of you know what CAR T, but may not have complete understanding. We will be touching upon that. And the two revolutionary tools have met together. Imagine CRISPR-Cas9 and CAR T both are revolutionary technologies in this era, and they together has brought an even impactful impact in science. So we will also see how these two revolutionary technologies are changing the field in drug discovery, challenges, and future outlook. So I'm sure all of you in your course by now you know the definition of gene editing, which is also could be genome editing, genome engineering. It's a type of genetic engineering in which the DNA is either inserted, deleted, modified, or replaced in the genome of any living organism. So this is a basic definition, and there are many different techniques in gene editing. So if you look at, there are 4 different techniques. The most old one is meganucleases. What meganucleases do is they can actually characterize a large recognition sites. I'm sure all of you know what's restriction enzymes, right? Restriction enzyme has a restriction site. They have certain recognition sites. So meganucleases, they can recognize a large base pairs of DNA, which is 12 to 40 base pair in the genome, which means that it will just occur once in a genome. So again, that particular enzyme recognizing another 40 base pair is very rare. So these are the meganucleases, which makes them very specific. And then the world has moved to zinc finger nucleases. What they are? These are again the artificial restriction nucleases. How these are generated? So they have a zinc finger DNA binding domain. These blue ones which you see is the DNA binding domains, and then the cleavage domain is taken from the FOC1. So these together, when it's fused, it's called zinc finger nucleases. So they go and bind with the help of the DNA binding domain onto the DNA double helix and bring down the cleavage. So that's how the zinc finger nucleases have emerged. And followed by that was talin. So talin is transcription activator like effector nuclease. Again, it's similar to zinc finger nuclease. Here, there is a fusion of TAL effector DNA binding domain and again a FOC1 cleavage domains. So this is even more specific. You can cleave any desired DNA based on creating a specific tails, I would say. So this is how tallins were emerged based on the specificity. And then to have CRISPR-Cas9, which was 2015 breakthrough of the year. And we will be going in detail for CRISPR-Cas9 as it has replaced these two technologies, though tallins are still used, but CRISPR-Cas9 has really taken over. And if you look into the cost, time, and also the feasibility, it's CRISPR-Cas9 which is more feasible. You can use it very easily. And a lot of specificity that you can get in CRISPR-Cas9 as compared to the mega nucleases era. Now, even if you compare all of these three gene editing techniques, I will not go much into detail. What you will have to remember is the advantages and disadvantages, which pretty much CRISPR-Cas9 has taken over. And so this is an old publication, which is 2019 here. So obviously the disadvantages, what CRISPR that's being mentioned here is now being addressed. So we are now in 2022, and within 3 years, these disadvantages have been addressed. So if you look into the zinc-fingered nucleases, it has not that great target efficiency. It's just 10 to 30%, and it's context dependent. As you see, the DNA binding domain has to go and bind onto the DNA duplex. And there are 3 domains in zinc-fingered nucleases. So the specificity of those domains so that they go and stay in tandem with each other, that is more important, which sometimes do not happen when you practically start doing the experiments. So high cytotoxicity, everybody knows cytotoxic, it kills the cells, and very complicated in designing it, because you see that there's 3 different domains, zinc-fingered DNA binding domains. So that's why that's a disadvantage. Talent. So here the complicated, though there is a good efficiency, again here it's high cost, difficult to operate, and it requires extensive sequencing, which is now taken over by CRISPR, which does give a good efficiency of 50 to 80%, very low cytotoxicity. The only concern that you always have to address is make sure that the target sequence it should have the PAM site. We will be looking into what PAM site is in very shortly. So CRISPR-Cas9 has enhanced both visibility, speed of drug discovery, and it will transform further. And as you see, the genome editing applications, it is in pretty much in every field. It could be in agriculture, gene therapy, cancer research, even the basic life science research. Every field, the genome editing has transformed, and very specifically CRISPR-Cas9. So if you look into the timeline of some of the industry-supported gene editing clinical trials which are happening, if you look into the timeline right from 2005 to 2020, so there are so many trials in CRISPR-Cas9, and if you see the light blue area, which is immuno-oncology. So that is what we're going to learn today. Immuno-oncology, you can see the CRISPR-Cas9 has been very, very broadly practiced in most of the clinical trials, and that is why it's important to understand how this has changed the drug discovery, specifically in immuno-oncology. So going into, before we understand immuno-oncology, I will briefly tell you what are the different types of cancer therapies. I'm sure all of you know about chemotherapy, right? I think one or the other in our own family is going through a cancer. Everybody agrees? Yes. So chemotherapy is something you've been hearing many times, right? And you see how the patients go through so much of side effects in chemotherapy. So in past 10 years, that is what has been very widely practiced. Though it is a lot of adverse effects, but there was no choice for the treatment. There are other therapies, as you see here, surgery. That is only the cancer has not spread, it's only localized, you remove that organ and that's where surgery is done. Radiation therapy, you use different radiations to kill cancer cells. Hormone therapy, there are certain cancers, when you provide hormones, it brings down the division. And angiogenesis, as you see that cancer cells, they grow very fast, they keep growing faster, right? Which means that so many blood vessels have to be formed for the cells, many cells to be formed. So what happens is angiogenesis is a process where the new blood vessels are formed, and you can actually inhibit those new formation of blood vessels, and that's what is called angiogenesis inhibitor therapy. And most importantly, now most patients are given immunotherapy, which is a cancer immuno-oncology. So we will address that. There are many leaps that has happened in cancer immunotherapy in a very short time. So right from 2004, you will see cetuximab, which was the first anti-EGFR agents. So if you look into this picture, that's your cell. You have growth factors outside the cells, which will help the cancer cells to grow. So you can use different inhibitors to inhibit these growth factors. So that's how some of the molecules will be designed to inhibit the growth factors that doesn't support the cell growth. And then now within the cells, you have different signaling pathways. And to target each of these signaling pathways, that's how different drugs are developed. So basic concept is, you want to kill the cell, it could be outside the cell, you block certain factors, or it could be within the cell, you want to kill some of the signal transduction pathways so that the cell does not grow anymore. So all the drugs that is designed, either it is small molecules or protein molecules, everything is designed based on these pathways and the targets that these cells carry. So cancer immunotherapy, it's an artificial stimulation of immune system. You're actually activating your immune system itself. Already you know when you fall sick, when you get fever, your immune system is activated. That's why your body temperature goes high. That means the immune cells are taking care. They are fighting the foreign body that has entered. It could be a pathogen or any kind of foreign molecules. So that time the immune system is already active. But what we are doing here in cancer immunotherapy is you're still more activating the immune system, and certain specific immune cells you're activating so that it can clear your cancer cells. So that is what is called cancer immunotherapy. And if you compare your normal cells, this is the DNA that is seen here, and in cancer cell there will be damaged DNA. So that is how they start growing uncontrollably. And if you look into your immune system, all of you are aware of adaptive immune system and innate. So innate is formed by NK cells. You have neutrophils, basophils, eosinophils. All of these form, and macrophages. All of these form the innate immune system, whereas the B cell, T cell actually forms your adaptive immune system. So we want to activate all these immune systems to clear cancer. So that is how cancer immunotherapy works. If you look into the tumor microenvironment, there are two situations that happen. One is elimination, that is your cancer cells will get eliminated. How does that happen? So you have your immune cells acting on it. You have in orange, that is natural killer cells, NK cells. It has certain receptors that will bind to the receptor on the cancer cells and it will kill the cancer cells. Or it could be brought about by CD8 plus T cells, that is cytotoxic T cells, that will also bind to the receptor on the cancer cells and release certain chemical molecules, that is granzymes, perforins, which will again kill the cancer cells. And CD4 T cells, where the dendritic cells, it will present the cancer antigen to the CD4, and this is how these three immune cells actually eliminate the cancer. But that does not happen in most cancers. You will see the cancer keep advancing. You will see cancer relapsing. So what is the reason? Because the cancer escapes from the immune system. So you see here, sometimes what it does is it will not express receptors on cancer cells so that the immune cells cannot bind. Sometimes it will express certain receptors that will engage the inhibitor receptors on immune cells so that the immune cell cannot act. So basically what you need to understand is the cancer can escape from the immune system based on certain mechanisms. And here those mechanisms are brought about by different receptors. So the two important immune cells that always takes care of cancer is NK cells and CD8 T cells. So there's so many receptors. So what you will have to understand is these cells, any cells that you take, they either have activating receptors or inhibitory receptors. So activating receptors will activate the cell, whereas inhibitory receptors will inhibit the cells. So that is how, based on the balance of plus and minus, your cancer cells will be taken care of. So cancer immunotherapy, if you look into what all has happened so far, first it started with only cytokine-based therapy such as interferon alpha for melanoma, that is skin cancer. Then antibody-based therapy such as trastuzumab for breast cancer. You have rituximab. So if you keep moving down, then came the immune checkpoint inhibitor, which was again a breakthrough in cancer immunotherapy. So what does immune checkpoint inhibitor do is there's a inhibition of immune cell by the cancer cells, right? So that blockage is taken away by these immune checkpoint inhibitors. So you have various examples. Have you all heard of anti-PD-1, PD-L1 therapy? Have you heard of those? Anyone? No? Okay, we will be addressing that. You will be so fascinated to see how these checkpoint therapy has gone beyond what it was. So there's something called trispecific antibody. So what does the trispecific antibody does? So all of you know antibody structure, right? In your immunology course, all of you are well aware of antibody. It has the two arms for antigen binding domain, and at the bottom that is Fc, which binds to the immune cells. It could be NK cells, macrophage, or dendritic cells. So you can design an antibody that can bind to cancer cells, that can also bind to immune cell, that is CD3, which you see in green there. It can bind to T cells, and the Fc portion of the antibody, it can actually bind to macrophages and NK cells. So what you saw, that one antibody is bringing together two different immune cells and a cancer cells, and look at the specificity. So now the immune cells can very specifically kill those cancer cells that express those antigens. So that is where the drug discovery and field has moved into in cancer immunotherapy. And we also have cancer vaccines. These are the names which is there, Gardasil and Provenge. And you have cellular therapies, which we will be discussing today, is CAR T cells. Why I brought in CAR T cells is the CRISPR-Cas9, the gene, one of the latest gene editing technique, has been applied in CAR T cells, which means that the drug discovery, drug development has gone to the next level to treat the advanced cancer patients. So you will be astonished to see how this has happened. So coming back to your monoclonal antibody structure, everybody now can recall the structure. Everyone at the back, you remember the antibody structure? Yes? All right. So if you see, there are a lot of antibodies. These can be from mouse, right? So the first structure, if you see, that is from the mouse, and these antibodies are actually the tongue twisters. These drug molecules are the tongue twisters. You heard me saying trastuzumab, rituximab. All these are actually the suffixes. You will see that it ends with omab, zmab, zumab, and umab, right? So what does this mean? It means that these antibody, their immunogenicity. So any drug that you give it to your body, you will have immunogenicity, that is reaction, right? Your body will react to it. If it's a foreign molecule, the body will react to it. and it cannot keep that antibody longer in your bloodstream. So it gets cleared. We need to avoid that, right? So you have to build a molecule that is closer to the human, right? So that is why you will see that if you go closer to fully human antibody, the immunogenicity is lesser. There will be no much side effects. But if you are at the other end, that is murine, that is mouse, there is high potential for immunogenicity because still that molecule is foreign to a human. So that is how the antibody drugs are designed, so that you will bring down the immunogenicity. And how the field has also moved further is you can put certain chemical molecules onto your antibody. What you see in the other end of the slide is these orange spots that you see. So these are the chemical molecules which you can attach to the antibody. Now you know you have antibody which you have designed for an antigen that is on the cancer. So now the antibody will very specifically travel and go and sit onto your cancer cell. Now, once it's on the cancer cell, the chemicals are also on the antibody. So the chemicals will also act on the cancer cells. So, which means an effective killing of cancer cells happen with these. It's called antibody conjugates. ADC structures, they're called as antibody conjugates. So this is another level of killing cancer cells, especially when it's spread too much and you need to bring down the tumor size. These kind of ADC structures are used depending on case-to-case situation. Because you don't want to use ADC structure when it's a mild cancerous case, right? You'll end up having overactivating your immune system as well. Because if there is overactivation, anybody knows what is an effect of overactivation of immune system?
Self-immune.
Yes. So there is one phenomenon that happens. It's called cytokine storming. So there is you have heard of storm. The wind, the storm, it keeps revolving, right? So similarly, the storm happens within your body. The immune system, immune cells are so abundant that it will kill your whole body. We will address that in the coming slide. So we have to be very careful in what dosage you're giving your antibody and what kind of therapy you're using in the antibody drugs. So you have been seeing those the Y shape of antibody, but if you look, when you make a drug of antibody drugs, it can be of any formats. You can use only one arm of the antibody as a drug. You can use only the upper two arms. You can use just a single chain variable domain where you have variable and one constant region, that is a variable fragment domain. Or you can do tri-specific, which I said before. One arm that can bind to a cancer antigen on cancer cell. The other arm can bind to a receptor on the T cells or any other immune cells. So this way you can actually bring different formats of antibody, and they now in today's era, these are the antibody drugs that are used in the market. So there's a huge potential, and even for COVID-19, which you have, I mean, we all are in this pandemic for past 2 years, the cytokine storming has been one of the major cause of death, not just COVID-19. It is cytokine storming that happens during COVID-19, and then the antibody drugs such as IL-6 had to be given. So you've heard of Biocon's IL-6 tocilizumab, right? Tocilizumab. So that was actually used for bringing down the cytokine storming in some of the COVID-19 patients. So those are the complications which COVID-19 also had got, and these antibody therapeutics came very handy. So you must be wondering how does this antibody actually function, right? So there are two functions. I mean, in any disease, it could be cancer or any other disease, there are two ways antibody are very critical. One is very important that the antigen binding site has to be very specific to recognize a specific antigen. Only then it will work, otherwise it won't. So you should have a specific antigen binding site onto your upper arm, and then at the bottom the FC region that has to be recognized by your immune cells. So how you see here in this cartoon, there are three mechanisms by which the cells die. ADCC, that is antibody-mediated cytotoxicity. Complement, all of you have heard of complement activation pathways. You have all the 3 different pathways in your immunology course. So that is another pathway where the cell gets killed, right? Membrane attack complexes form, pores are formed on the cells and they get killed, right? So that is one way the cancer cells will get killed, and the other way is phagocytosis. So ADCP. So in any industry, if you are into this kind of job role, designing in vitro assays in the drug discovery, so these are the common mechanisms of the drug by which the cancer cells or any disease cells that you're intending to kill, this is how the different mechanisms take place when you design antibody therapeutics. Now we will come to our important topic, that is T-cell receptor. So it looks same like an antibody, which we just studied, right? So if you see the T-cell receptor, it has alpha and beta chain. It has CD3 co-receptors, and you also have a cytoplasmic tail. And the one that is embedded here, that's called transmembrane domain. So this is T-cell receptor. And if you see how CARs are designed, CAR T cells, I'm sure you've been hearing about CAR T so much. Pay attention to this slide because it will help you clear your and you will understand what is CAR T actually. So if you see on the upper cartoon, this is antibody and the lower is T-cell receptor, right? So how CARs are generated? The first portion, that is the variable region, single chain variant has been taken from the antibody because that can bind to your antigen on the cancer cells, right? So antigen binding domain is present on these tips, right? So that part is taken and it's got fused here. And you have a transmembrane domain from your T-cell receptor, and then also the cytoplasmic tail, the CD3. So this entire construct is called CAR. So everybody got it? So we will now see further how these CARs have also evolved. So what it does basically, it will behave like a T-cell receptor, but now these T cells can easily go and attack cancer cells because they have the antibody arm, because they have those antigen recognizing capability now. So that is how these, it's called chimeric antigen receptors, which are being developed. And how does the T cells kill? All of you going again back into the basic immunology. So we have been studying so much. When I was like you, I used to by heart, okay, CD4, MHC. I'm sure all of you do that, right? I was also doing that. MHC2 means yes, it is CD4. I would, try to memorize it so many times. But if you really understand the mechanism, you don't need to do it anymore. So here is a slide which will tell you clearly that how does the two types of T cells come into play. So you have this T cell, that is immature CD4 T cells. And what happens, always remember, the lesser the number, CD4, it will go and bind to the MHC2. So when it binds to MHC2, it recognizes the antigen on MHC2. CD4 T cell responses are exhibited, that is Th1, Th2 response. And when you see that CD8 T cells, it binds to the antigen presented by MHC class 1 and it brings about cytotoxicity, that is, it kills the cell. This is very important when, especially in your drug discovery, when you actually you're trying to understand what is you have manufactured your drug, but how are you going to say how it works in the body? What is it going to do, right? Until you do not define what your drug will do, how it is going to kill your cancer cells, the FDA will not take the drug forward, either in phase 1, phase 2, because you need to know how the cancer cells are being killed. The specificity have to be defined. If you do not define, it's good for you also. It's good for patients who undergo clinical trials. They will not have side effects. And why it is good for you? because your drug will not fail. If you're sure that this is the pathway, my drug that I have designed, it's going to kill through this pathway, you are very sure the drug will not fail in future. Whatever you invest in your drug discovery and development, you can save that money and it is worthwhile, you can earn back. Especially, that's why it's very important for companies when they invest, mechanism of action of any drug has to be defined. And that is why understanding these processes are very important. So let's see now what are the different ways the T cell activation happens. You have T cell receptor, you have, that is first, one is the binding, the second is co-stimulatory interaction, you have cytokine signaling, cytokines are released and cytokine signaling happens. So this is how the T cells get activated. Now in CAR T cell therapy, we just learned how the CARs are designed, but let's see in therapy what actually, how it actually works. So what they do is they isolate the T cells from the patients, right? You take the T cells, you draw the whole blood. There will be different procedure to only extract T cells. So you take out those T cells and then you genetically engineer for it to express the CARs, right? So what you do in the second step? You're engineering those T cells after you've isolated T cells from the patients. And once you do it, the genome editing, it could be any technique, but mostly it will be CRISPR. I will discuss in the coming slides. So T cells with now CARs are expressed on it. Now these T cells are put back into the patients. So these CARs now can go to those target cancer cells and they can clear the cancer cells in the patient. So this is how it is done. But there is a drawback, right? For every patient, it has to be engineered and put back. So the field wants to have, can you design something that can work for whole population? So you want some CAR T cells. You don't want to every time drop the patient, keep giving a specific delivery of the T cells. You want to design something that can be universal, that can be delivered to all the patients in the world. So that is how the field has now moved into develop a universal CAR T cell therapy. So this is how it is. Can you, if you can just see, you are drawing the blood and you isolate the T cells, you make the CAR T cells, you grow them in your cell culture lab, you're growing a lot of T cells, and now you are putting back those CAR T cells into the patients, which will now go and kill the cancer cells. So this is about CAR T cells. Everybody is still with me? Hope it's not too heavy onto you. I've simplified it. It's a complicated topic, but trying to put it in a language that you can grasp it. Yeah, so stay with me. We will now go into CRISPR-Cas9 technology, the latest gene editing technique which is being used in drug discovery, which has transformed drug discovery. And one specific case that we're going to use today is the CAR T therapy. So everybody knows about our immune system, human immune system. Now let's go into bacterial immune system. So if you look at into, again, bacteria has innate and adaptive system, and CRISPR-Cas9 system is actually bacteria's adaptive system. So we have used bacteria's adaptive system in gene editing technique. So let's see how that was done. So its CRISPR journey, it started with an observation. The scientist, a Japanese scientist, back in 1987, he saw that there were some mysterious repeats in the bacteria. There were like chunks of repeats when he was observing this bacteria. He was wondering, what are these repeats? It looked fascinating to him. And then he got to know it is palindromic. Everybody knows what is palindromic, right? So if both the sides, it reads the same. So he saw that they were repeats and they were palindromic repeats. And they are like clustered, you know. Similar observation again was made by another scientist. That scientist, he just made an observation, but he could not know what are the role of these repeats, how it is functioning and why it is present. And then similar observation was made by Francisco. He saw it in another bacteria. He also saw these unusual repeats. He was wondering, what are these repeats? Again, these are palindromic. Again, his curiosity increased now. As scientists, I think curiosity is what makes you as a scientist. So, when the more curious, the more breakthroughs and research that you bring to the table. So, he also started seeing there's something between these repeats also. And these are called spacer DNAs. And he wanted to understand what are these spacers. And the best way to understand any DNA sequence where it belongs is doing BLAST, right? All of you also have learned bioinformatics too. So what you do by BLAST is any sequence that you have, unknown sequence, you can just put it into the BLAST. It will go and match to which organism that sequence belongs to. That way you will get to know where that sequence matches to. And he did the same. He was not sure what is this, so he did a BLAST. And then he found this sequence belongs to a virus. That was what he found out. And he wondered why it belongs to a virus. And these are called Cas genes, that is CRISPR associated genes. So you see those, there are spacers and there are repeats. So this entire the entire frame is called as CRISPR-Cas9, and they belong to virus. So why did that happen? And let's dig more deeper now. So Cas genes, they encode for Cas proteins. There are two features of this Cas. One is a helicase activity, that is unwinding your DNA. And second is endonuclease activity, that is your chopping, cutting your DNA. So these are the two properties of this Cas genes. And again, scientists figured out that this CRISPR-Cas9 is a part of bacterial immune system. As I mentioned, it belongs to adaptive immune system, and it actually helps the bacteria to fight the viral infections. So you know bacteriophage, right? Most of the bacteria, they get killed through virus, that is bacteriophages. So when bacteriophages attack bacteria has to activate its immune system to clear the bacteriophages. So that's where the CRISPR-Cas9 was helping the bacteria to clear the viral infection. And let's see how did they do that. So there are three-step protection mechanism. So first, the bacteriophage is now onto the bacteria. Once it's there, it will empty its DNA into the bacteria. So you see that it is releasing its DNA. Now this green portion you see, it goes and gets integrated into the bacterial genome, right? It's gone into now integrated into the bacterial genome. And what it does next? In the next time, when the same bacteriophage infects bacteria, it already has that DNA inside it. So what it will do is it will go and clear this along with the Cas9. It will go and attack the bacteriophage DNA, and it will no longer allow it to infect. This is the defense mechanism it evolved, and that's how these CRISPR repeats got inserted, and they were from the bacteriophage, and it's the self-defense mechanism. So this is how there are three steps. Once the virus releases its DNA, it gets integrated into the bacteria, and when there is a second time infection, it will recognize that with Cas9, it will go and chop that. So that is what even our immune cells do, right? You've heard of booster therapy. We take a lot of COVID-19 vaccine. All of us are in the line to take boosters, right? So what it does? It's basically you've taken your primary dose. You want that memory has already remained. Now you're taking a booster dose to build that memory again, to bring back that memory, so that all the time your memory cells, your memory immune cells are there in your body. Whenever the next COVID-19 comes and attacks you, the memory that is there in your immune system will clear that COVID-19. But unfortunately, the COVID-19 is evolving. There are different variants. So it's all about how well your immune system can recognize the different variants in the COVID in the COVID-19, right? If it's a variant and if your immune cell no longer recognizes those variants, it will not clear. So that is how, the variants evolve, and our immune system can no longer attack those variants. But there is a solution, some region which is very common, even among the variants or the generic COVID-19, let's say. If there is a certain region which is very constant, you could still have an antibody design or a drug designed to that particular region and the therapy continues. So these are the different protection mechanisms which bacteria evolved, and this is the CRISPR-Cas9 system. And we will now look into how this defense system became a gene editing tool. Just imagine, it was a defense system in a bacteria, and now we use it as a tool to do all kind of discoveries and all kind of applications. So, as I said, it was observed in bacteria and also in archaea. So these are the two scientists who thought that why not we use this as a genome editing tool? Why don't we bring it into the labs or even in drug discovery? So what they did is they took that portion which binds to the DNA, which is called as cRNA, that is complementary, and then it was fused with the tracer RNA. So now this is called the guide RNA. So what it does, you have the guides which is very specific to the DNA, the blue portion that you see here, this portion. So this has to bind to your specific DNA of interest. So you can keep designing different sequences here. Based on that, it will go and bind to your DNA, and then you can use this system. So let me show you here. So you see that this entire portion is called as guide RNA. So any genome editing that you want to do with CRISPR, you have to be very sure of how you need to design your guide RNA. And these are the two scientists who got Nobel Prize in 2020 for developing this method, and it's now in use. So let's look into what are the different components and what exactly it is. So as you already know, based on adaptive immune system of bacteria, this technique has evolved, and there are two components, that is guide RNA and you have Cas9 system. So these are the two components of CRISPR-Cas9. So what it does, guide RNA, it will go and bind to the specific DNA sequence, and the Cas9 is an endonuclease. You see that it unwinds and it's an endonuclease with chops. So Cas9 will come and bind there and it will cut your DNA. So you saw that, that is how it is. And now once your DNA is cut, it has to be repaired, right? There are two ways your DNA can get repaired. Either by NH-EJ repair mechanism, both are double-strand DNA break mechanisms, or homology recombination. So here index, what it means, insertions or deletions happen when there is a repair that happens. We call it as index, insertions or deletions. Whereas in homology repair, there is a insertion that happens, that is knock-in. So these are the two ways your double-stranded DNA occurs. So concept is clear. CRISPR-Cas9, two components. You need guide RNAs that will specifically go and bind to your DNA of interest where you want to cut or chop that particular gene of interest. You have Cas9, which will cut the DNA, and then the repair machinery will get activated. Either of these two mechanisms happen and it will repair your DNA. So now let's see, bringing you back into the cars again. Everybody is still with me? You need some stretching? You need some stretching? No? You want to stand up a little bit and stretch? No? You're good. Okay. We'll take that break in a few minutes. So, cars. Bringing you again back into the car. So now you know you have a car created. Now we're going to apply CRISPR-Cas9. CAR, you saw it's an advanced level in CAR T cell therapy. Now we're going to add CRISPR-Cas9 into the CAR sandwich. Okay, let's see how we make that. So again, this was introduced by scientists at University of Pennsylvania. It's a revolutionized cancer therapy, and there are two FDA approved CAR T cell therapies. You have Chimera and you have Yescarta. These are the two approved therapies. If you see for which one it is used, it is for acute lymphoblastic leukemia and the B cell lymphomas. So these are the two CAR therapies, and when there was success for CAR T therapy, there was more in search of how this can be applied to different cancers. Like there were two cancers where the CAR T cell therapies were applied. Now the research started that how we could apply the same therapy for different types of cancers or even different diseases, right? So if you see, there are challenges with CAR T cell therapy. So whenever we invent or discover something, there's always challenges or there's always disadvantage that we will find, right? So even in this technology, there were a lot of disadvantages. So if you see that what are the issues with this? You have cytokine release syndrome, as I said, cytokine storming. So that is there with CAR T cell therapy. And there's something called anti-CAR antibody that is being developed. What do you mean by anti-CAR antibody? That is, an antibody is developed against your CAR itself, which will kill your CARs. So that is another challenge. Can you see how complicated it is, right? So, and then the second is lack of persistence of CAR T cells. So CAR T cells have to be maintained. When you put it back into the patient, the cells have to persistently be there to bring out the action. But keeping that numbers is also a challenge. And then it will relapse, right? Sometimes the CD19 when you make anti-CD19 antibody. So there is a relapse of the epitope, the change of the epitope. So in all of these cases, the CAR T cell therapies would work, but there is again a strategy how it can be brought about. So cytokine storming, as I already said, tocilizumab from Biocon, this one actually is very well used during COVID-19, as I mentioned. So what it does is IL-6 is the one that overactivates the immune system. You need to block IL-6. So IL-6 is blocked by this antibody. And for anti-CAR antibody, right? So for that, you have to use a humanized CARs. Like I said, the fully human antibody, if you choose, you can bring down the immunogenicity. So anti-CAR antibody is mainly because of immunogenicity, right? If your CARs are more, your CARs are not actually fully close to the immunity, it is considered as a foreign body. That's why the antibody is generated against CAR. So another strategy is to humanize the CARs. And lack of persistence of CAR T cells, you could do that by different ways. I'll not go into details, it's not needed here. And we will now look into, so here for CD19 epitope escape, you could actually do different target. There are different other targets on the same B cell lymphomas, so that can be used for targeting. So you will now understand very clearly how this checkpoint inhibitors are used and how CRISPR-Cas9 is being used to overcome the potential challenges of CAR T cell based therapy. So these are the 4 challenges of CAR T cell therapy. So everybody can see that cartoon. You have T cells and you have tumor cells. So what happens? PD-L1 is a receptor on the tumor cells and PD-1 is the receptor on the T cells. So when these two interact, the T cells can no longer act on your cancer cells. It is a blockage. It's not allowing, it's a negative signal, inhibitory signal. It's not allowing your T cells to act on the tumor cells. And you need to break this. So how do you break it? You need something to break this interaction. So either anti-PD-1 antibody, so which is here, or you generate anti-PD-L1 antibody, which is here. So when your antibody binds, they will no longer interact. And now the T cells can act on your cancer cells. So this can be some disruption of inhibitory molecules and signaling axis. So you can disrupt this PD-1 expression through CRISPR-Cas9. You can reduce CRC and GVHD. So GVHD is the graft versus host syndrome. So you know that when you put a foreign, let's say organ transplant, right? When you want to transplant your organ into another patient, you take from a donor, you put it into the recipient. So does the patient take it up? If it's not matching closely, the HLA matching doesn't happen, they will reject. So you will hear a lot of like organ rejection, transplant rejection. So that is what happens even in CAR T cell therapy. When you can no longer use the same T cells from the patient and inject back, but you want to use, let's say, a universal T cell, right? It will get rejected because it's coming from a different donor. It's not going from the same patient, right? It's an allogenic T cell. It's no longer autogenic T cells. It's coming from a different donor. Now when you put T cells into the patient, it will get rejected. So that is a major challenge, and this is also overcome by CRISPR-Cas9. So basically, we want to manufacture universal CAR T cells, and also TGF-beta, this is actually an immunosuppressor. Whenever you've heard of activating immune system, right? But there are also few factors which will bring down the immune system. One such factor is TGF-beta. You've heard of regulatory T cells. You saw CD8, CD4, but there's something called regulatory T-rex, they are called. And these regulatory T cells will suppress your immune system. And that happens again in CAR T cell therapy. So all these issues are addressed by CRISPR-Cas9. So this is the TGF-beta which I was telling you. So you have this solid tumor, you have CAR T working on your tumor, and the T-rex, if you see, it will go and inhibit your CAR Ts. It's bringing down the effect of CAR T, so you need to block the T-rex. So again, we will see how all these can be, these challenges can be overcome. So this is the cytokine release syndrome which I was talking about. Your body immune system is attacking your own body, and see how when your body attacks you, how those blackening is happening. Though it looks like as if it's a fungal or some bacterial infection, but it's your own immune system that's attacking you. And this was first observed in one of the treatment, that is this theralizumab, which is anti-CD28 antibody. So what happened in this case is this company, Paraxel, they developed this, and it is for the receptor CD28, anti-CD28 antibody. Everything was done in drug discovery. You will actually do preclinical trials, right, in animals. Everything is tested in animals before it goes into humans. So in animal, what you will understand, whether it is toxic. So there was no toxicity, and even the dosing, you will understand how much amount to be used in the animals, and you do so based on animal data, the dosing is decided. That dosing gets it's elaborate. I would say extrapolated for the human dosing. In this case, even they took a 500 times lower the dose that was tested in animals. Still, when they went to the humans, there was this kind of adverse reaction. So this is one of the adverse reaction of antibody drugs, and this has to be addressed, which happens very commonly in CAR T cell therapy. So that has to be addressed, and we'll see how that is being addressed. So CARs itself, as I said, it's revolutionized, and you see there are so many generations of CAR that has come. We have first generation, and you see how those changes have happened. Here, one co-stimulatory region was added. Here, 2 was added in the third generation, and there were also certain cytokines which cytokine targeting CARs which were generated, and in the 5th generation, CRISPR-Cas9 is being utilized. So right now the era is in the 5th generation of CARs, and CRISPR-Cas9 has transformed the speed. Let's see how it does. So everybody understood checkpoint inhibitor, PD1, PDL1. So that has to be in the tumor cell. So you will understand. I'll just take you quickly through one of the research papers so that I want you to understand in realistic how they do discovery and how the drugs are proposed. So this is one of a classic example. So you see the blue is the T cells, and you have pink color, that is tumor cell. Everybody with me? All are able to understand? So now the T cell has this PD1, okay? And your tumor cell has the CD19. So the CAR that the T cell, this is a CAR T cell, right? So the CAR T cell has CARs for CD19. CD19 is your tumor antigen. So now you want your CAR to bind to the tumor cell so that the tumor cell gets killed. So you will see that you are designed a CAR T cell that recognizes the CD19 on the cancer cell, and the expectation is it will clear the tumor, and that's what happens. And you know that here in the previous example, let me take you back. So you see that PD1, PDL1, if these two interact, cancer cells will not get cleared. You need to break this interaction through these two antibodies, right? But in CAR T cell therapy, you don't want to use antibody also, right? It's a cell therapy. You can't do so many combinations. Putting antibody drug also, putting CAR T also, it's not feasible. So what we do here, what they did here is now you want to delete this PD1 so that it will not interact with this PDL1. Because if PDL1 is present on the cancer, which is the situation most of the time, the tumor will not get cleared. Okay? So now these CAR T cells in which the PD1 has to be removed. That we will see in the next subsequent slide. But let's see the effect of having PD1 alone on T cells and then the PD1 PDL1 interaction. So you see in the figure B, the cancer cells which had PDL1, you see here the cancer cells which had PDL1, the degranulation. So what is degranulation? That is, your cells will release the granules out, which means that your cells are getting killed and the granules are released out. The cellular contents are released out. So if you measure those degranulation, it's an indicator of cell death. Okay, so that is what happens here. So whenever there is PDL1 present on your cancer cell, the cancer cell death is lesser as compared to this gray bar, which do not where you do not have it in conjunction with the cancer cell that has PDL1. Okay, so again, cell killing mostly brought about by CD8. So you will see that the cancer cells which had PDL1 here, it's not dying that faster, whereas compared to just that had CD19. Similarly here, right? So even in the mouse, they have studied that the tumor clearance was much better when there was no PDL1. So this is one of the experiment where you could this is an evidence that PDL1 PD1 interaction will not let the CAR T therapy work in a way it has to. So for the CAR T to work, it has to be removed. Now, even in this graph, you will see tumor clearance. The tumor, this is the y-axis is tumor burden. So wherever the cancer cells that had PDL1, the tumor is growing, it's going upwards. Whereas the others which did not have PDL1, they are dead with the CAR T therapy. So similar, this is the survival graph. You will see the cell death here. Now, CRISPR-Cas9 is used to delete this PD1 now. Because as I said, you cannot have CAR T plus your checkpoint inhibitor as a therapy. You want to now endogenously remove PD-1 from the CAR T cells. So that is how you see that this is now removed. So first is PD-1 editing, then you are going to inject those CAR T cells into the patients. And this is the flow cytometry. I'm sure all of you have heard of flow cytometry. It's one of the techniques to understand the cell surface expression markers. So through that you can understand what kind of markers are expressed on the cells. And this way, when you do a lot of editing on the cells, this is another way of validating whether you've got the right cell population. So through flow cytometry, you can see that we're going to sort the CAR T cells which do not have PD-1. Now only those CAR T cells you're going to use it in the patients. So how do you sort out those populations? Through flow cytometry. So there are two axes. One, the y-axis is PD-1 expression and the x-axis is anti-CD19 expression. So your CAR T now has CD19, like anti-CD19 receptor that has to go and bind to your cancer cell, right? So you have to make sure your CAR has that anti-CD19 portion, and you no longer want PD-1. So if you look into the y-axis is, these are the cell population which is boxed down. These are the cell population which are CAR plus, that is anti-CD19 positive, whereas PD-1 is edited through CRISPR-Cas9. So no longer it has. So this will be now used in the therapy, a universal therapy which I was talking about. So this is how you can manipulate the genome within the T cells and generate CAR T cell constructs to be applied to different patients where the PD-L1 is expressed, which is the general scenario for all the cancer cells. And a universal CAR T cells can be generated using CRISPR-Cas9 technology, and we are now in the era of triple genome editing. It's very fascinating, and let's see where the technology has gone and what is that triple genome doing. So this is again the T cells, the CAR T cells. Using CRISPR-Cas9, we just saw how PD-1 was deleted. So that was here, the inhibitory receptor PD-1 was deleted, and you already have CAR. Let's say, for example, that CD19, any domain that recognizes antigen on the cancer cell, you have put that. You don't want HLA-E and endogenous HLAs. As I said, the host versus graft, the organ transplant rejection. You heard that. So there are some HLAs which are present on T cells. They will get rejected when you inject it into the patients. So you don't want the CAR T cells to be cleared from the recipient's population. So you want to knock down the HLA classes. And endogenous T cell receptor. You don't want the endogenous T cell because too many T cell receptors, cytokine, storming. So you just want a specific T cell that is CAR receptor, only that pathway or that signal to happen on the cancer cells to come in action. So you saw three things, triple genome editing. One is CAR, specific for your recognition of your anti-cancer cells. Second, PD-1 deleted. Third, HLA deleted. So this is now the universal CAR T cells, which is in trials. A lot of trials are being conducted, and this is generated through CRISPR-Cas9 technology. So if you see, these are all the trials, and they're already in phase 1. Some of them in phase 1 and 2. So this is how CRISPR has evolved. As we learned just a bacterial defense system, it became a gene editing technique, and from the gene editing technique, it has gone to a triple genome editing technique. So you can see how it has transformed drug discovery or even treatment in cancer, right? So all of you could understand where these two revolutionary technologies, both CAR T cells and CRISPR-Cas9, have changed the paradigm of treatment in patients. However, some of the technology. It's easy to use, simple design and cost effective also. It might show few off-target effects, but your guide RNA designing is very important. These days, even at Merck, we have a studio where you can do a guide RNA design. So when we do a lot of our hands-on trainings and also our digital workshops in CRISPR-Cas9, we teach you actually how to design those guide RNAs using those softwares, and we will also make you do those experiments in our lab, so you can actually design the guide RNA for yourself for your gene of interest and carry out the CRISPR-Cas9. So as you see, there are slight disadvantages. You need to have those PAM sequence. So PAM sequence, why? Because your Cas9 can recognize only when there is PAM sequence and it will make the cut. So that is why having a very specific PAM specific Cas9s are important. And we do a lot of computational prediction programs. As I said, we have the studio to design all of the guide RNA. So I think if you take up those workshops in detail, I can go through how we design those. Yeah, so I think I will stop here before the entire technical questions. So this is the entire journey of gene editing technique and how it has changed a lot of things in drug discovery and treatment of patients. Anybody has any questions? Hope I did not put you all to sleep. Still active? Some more noise? Good. So anybody has any questions? We will be stretching now, but if you have any questions, I can take those, or we can move to the second part and take the questions later as well. Someone has any question? Everything was clear? Yes, please. How the culturing of CAR T cells? Okay, good. So this comes under mammalian cell culture, right? So it's a cell biology technique, mammalian cell culture. So do you have cell culture facility here? Have you tried to culture any of the mammalian cells by any chance? Okay. So what you do is we in the cell culture, just like a bacterial hood, we have a mammalian cell culture hood. So you have certain media, which is called, for example, RPMI. RPMI is a media for cell growth. So when you're growing cells, it has to multiply every day, right? So let's say you have some 10,000 T cells that you took from the patients, and you have now put them into the media. And then when you've taken it from the patients also, it takes few days to adapt to the laboratory condition, right? Bringing from the patient, the environment of growth was much different. Now you're bringing it into an artificial condition where you're growing it in your laboratory. So you provide all those components, media components, which are similar to what is physiologically present. So you grow those T cells every day. So when you have 10,000 cells, T cells have a doubling. Every cell has a doubling time. Let's say some cells divide after 24 hours, some 48, and there are cells which will divide after 62 hours also, or 72 hours also. So based on the doubling time, you will have to culture the cells. So if it is 24 hours, you get good population in a week, you can reach to millions of cells, millions of T cells. And that's when you have enough population, you do CRISPR-Cas9, and then you do selection of the mutants. So I did not go too much into the details of CRISPR-Cas9, how to conduct in the laboratory experiments, because the topic was more towards application in drug discovery. So these are the technique where you can do screening of your CRISPR mutants. So that is how I hope I answered your question. The T cells are cultured by providing the same physiological conditions in the laboratory, and based on the doubling time and how much cells you want to grow for your own application, you keep growing those cells in the laboratory by continuous supply. And very important, cell counting. Right, when you need to be sure how many cells, because when the cells get over, T cells are actually suspension, so they can still stay. in a suspension. But when you have other mammalian cells like cancer cells, like PC3 cells, they grow as a monolayer. If there is no space for the cell to grow, they will start dying. So there's a technique called trypsinization. That is, you trypsinize the cells, you break all those cells which are attached to each other, you singularize them, and you do a counting which is called hemocytometer is a technique where you do cell counting. You stain the cells, you count it under the microscope. So hemocytometer is a technique, you do cell counting. So providing media, understanding the doubling time, counting how many cells are being produced. So 10,000 cells, if the T cells has 24 hours as a doubling time, in 24 hours the 10,000 becomes 20,000. In next 24 hours, the 20,000 will become 40,000 because they're doubling. So based on that, you can take how much ever population you want and use it for your application. So you can perform either CRISPR or you want to put it back into the patient. So that's how it is done. Yeah, clear? We'll go to the next. Okay. Out of time, so let's go to the next. I think before we get started with the next part, which is very, very important to all of you, let's do a little stretching. How about doing little stretching? Pat your friends back. Yeah. Say you're awesome. Okay, don't pat too hard. All right. Good job. All right. So we will move into the next part of career mapping. So how many of you, I want certain volunteers. How many of you have already decided what you want to become in next 5 to 10 years? Anybody decided? Nobody has decided. There's a lot of time to decide, right? Who cares? When the time comes, we'll decide, right? So, all right. So let's address that because it's very, very critical for you. If you do not decide what you want to become, you will. Yeah, I think we want some silence from the back seaters at the back towards my right side. Yeah. So I request you to pay attention. So apart to move forward, I'm sure all of you, irrespective of what you are doing currently, either you're in bachelor's or you're doing your master's, final year, first year, whichever year you're pursuing, you have this question, what is next? I have this question even now, what is my next? What am I going to be next 5 years, right? So your career, it's a journey. There is no destination. So remember, you have to always work hard for your career. There is never an end. It's a journey. So have that mindset right at this age. If you do not have that mindset, you lead into a lot of disappointment or I would say discouragement. You'll be discouraged that, oh my God, I have to keep working. But it's not that. Have a mindset that it's a beautiful journey. It's a journey where you become better, right? So I had the same question, taking you back into my journey. I did my biotechnology, though I was being introduced already. I'm bringing it back because I did not plan for my journey. What I am today, I definitely did not plan. It has happened, but my decisions have definitely helped me to be here. So this entire session is for you to be aware, to make you aware of what is available to you, and also to help you take decisions for your career. So that is what I would like to do in these sessions. So I did my BTech in biotechnology from SJC. And as I said, I again wanted to ask myself what next. As doing bachelor's, it's always important to pursue your master's, I would say. Because there are a lot of people who have entered into industry just after bachelor's, and I'm not discouraging, but when you grow up the ladder in your role, either in academics or industry, the credibility is always seen in terms of what degree you have, right? So it's always good to have masters, which will help you move forward. Or maybe if you're doing bachelor's and you want to do some some other degree you want to add on, maybe some online courses or some other you want to pursue and add on, which is also good. But masters, I would say, encourage you all to do, which I also did at VIT Vellore. I did my PhD. I was really not sure of doing PhD. I did not even plan to do a PhD. But when I was at the final year of masters, I did my project in IIC. And as you know, IIC is a research-rich environment. I was inspired. So most of you, what as a human being, we get influenced by our surroundings. It's very important who are your surroundings. It could be your friends, your family, your professional network. When you have those environment, it will influence your decisions. So that happened to me. Because I was in a research-rich environment, it influenced me to do a PhD. So I went ahead and did PhD, but though I was thinking, no, I should do an industry job, I should go to an industry. But I was always influenced because of my environment, and that is very natural, that happens. And I did my PhD, and I went ahead to do a postdoc. Again, I was still carrying that question that no, I need to go to industry. I do not want to be in academics. But I started realizing that most PhDs, they go into doing a postdoc. I think that's the trend, traditional trend that happens. Though I was trying to avoid, I couldn't, and I went ahead and did. But I also thought I made this decision based on, see, when you make decisions, you consider your personal situation, you also consider what's important for your profession. So I took this decision because I had just delivered my son. He was just 10 months old, and I thought if I go into an industry job, it's going to be a lot for me. So I thought maybe doing a postdoc will give me that time to take care of my son. So again, my decision was driven by my personal life. So so I went ahead and did my postdoc, and then I think finally there was a dream that I want to be in industry. I'll tell you for two reasons why I am in industry. Because I felt especially in academic research, I would say, not really discouraging. It's everybody's choice is different. So when I was doing the research was very slow. The application that you bring onto the human health takes longer time. So this was my personal choice that obviously I have the dream that I want to be in higher positions in industry, and having a PhD and postdoc experience will directly put you into higher positions in the industry, which happened to me. Which doesn't mean that you should do a PhD or a postdoc to get into industry. It doesn't mean. It's all your personal choices. What is suitable for your current situation and what is your vision and your passion for your career. So that is what the decision that you have to make. So I joined Zomato Biologics as a scientific manager, and then what happened to me in 2021, I joined as commercial marketing manager. So I switched from a hardcore R&D into a business now. Like business, what I mean is there is partially science, there's partially business. So science, wherein I would say the labs which we have, we do a lot of research collaborations. I always want to stay closer to science because I have invested 11 plus years in science and I don't want to go away from science. So I wanted to make a choice where I can be closer to science. And I was interested in the business part also because I do not wanted to end up doing a lot of bench work all the time. I wanted to actually move ahead into the higher role. So business as in, how well you can satisfy your customers. The business is all about what is your offerings, what you want to offer to your customers, and that is what it is for our labs. We help, our offerings is we help in skill development programs, research collaborations. That's what we do. So this is my journey. Why I shared is it was unplanned, but cautiously the decision was made. It was me who took the decision. It was, though I sensed what was going in my environment and I took the decisions. and today I'm here. So I want you all to have something similar, and you have to become what you want to be, not who others are. You don't need to be like what others are doing. So you just start following their path, and you might not even be interested in their path. That will happen only when you get into that path. You'll realize only when the other side of the grass is always green. So start watering your grass. That is what is needed. So here I'm using a situation like be like a tumor cell. The tumor cells are not good, but I'm telling you, be like a tumor cells. Keep evolving in your career. Keep building your best versions, and that's how you will reach to the epitome in your career. So to go ahead, I feel very blessed that I have an extraordinary journey. So when I was doing my postdoc, I used to do, research work. It comes with a lot of frustrations as well, because you want certain things to work in certain way, and the experiments fail. And also I had my son. I had put on weight. I wanted to get in shape. So I thought I should start some exercise. I should get back to my shape. So I started running. So I first signed up a 5 kilometer run in one of the event. I had lost some weight. I thought, okay, I can do a 5 kilometer. So I'm bringing out this example so that it could touch some of your lives too, because I am not what I was 4 years back. So when I signed up for this race, this is something I have never done in my life. It was a 5 kilometer race. So I thought, let me just go do it. Like, we also train it. Very important. Preparing for something is always, for your exam, you need to prepare. Only then you will excel. People who prepare will definitely have success. People who will just clear, even if they're not prepared, it just happens as a fluke, one or twice, but not always. So I finished 5 kilometer, and it gave me a lot of confidence that, oh my God, I could do a 5 kilometer, which I did not even dream of doing, and it felt like I'm so powerful. It's so good. So I went on to do 10, 21, 30, 42 ultra marathons, 50, 60 kilometers. So I'm a marathon runner. I do 40 to 60 plus kilometers. Last year I did 2,300 kilometers for the entire year. And so that is it. So just a small decision or an action could bring me this much of success. I never thought I'll be a marathon runner in my life. And then, as I said, this is a new skill that I've developed. So long distance running is a skill, because you have to maintain your cardio for a long time. So everything that you do is a skill, and this is a new skill which I learned myself. So when you are a PhD, you keep inventing trial and error. I did the same with running as well. Coming from using my PhD skills, I did a lot of trial and errors, and I understood my own body, how it is adapting for the runs. And nobody coached me. It was me who trained myself. And I'm proud to say I'm a marathon runner, and these are my medals so far, and there are many more. I've not taken the latest picture. And then when you have skills, when you know how to do, and your experience that you get, you can share that experience to anybody. So that experience will help them to get better, or that experience will tell them to become success, right? So I felt that me, though I was not into fitness, though I was not a runner, could become a marathon runner. Why not and there was a lot of change in me that was happening. I became very confident, and it was adding fuel into my profession also. I was doing my productivity at work also increased. I was healthy, I was very positive, the relationship also improved. So when you become better, everybody around you also become better. And whatever you take up also, it becomes fruits. So the second layer was, I thought, if I have to say give that knowledge to people, they would think, what does she know? She just ran something, and why is she coming and teaching us, right? So obviously you need some credibility. Even when you go for the job, when you say I have done my master's, but they will ask for a certificate, right? Show me your master's certificate, or show me that you have gone through this workshop. The certificate, honestly saying, it's for just showing. But actual thing is what you know and what you bring it to the table is actual. So a lot of people might have certificates, when they go on to the job, they fail because they have not really done that. So I went ahead and did a lot of certifications in fitness. So I am actually a personal trainer in fitness. I also got certified as group fitness instructor. I actually teach a lot of in the gyms in Bangalore, such as Gold Gym, Apple Fitness. I teach group classes early in the morning. Then I go to work, and some of the days I finish my run, and then I go to work. So I enjoy doing this. This is the life that I think I wanted to be, and this is my destiny, which I truly feel. And I have my own running club now. So that's my son with the yellow shirt here. So I have produced 40 runners, all non-runners. So if you see that my running club name is called Running Genes. So it's not Jeans, it is Gens. So which means that the science and the fitness is my passion. I'm pursuing both of these passions very equally, and fitness has become my second career. So I even earn through fitness. I have running membership. I'm certified. I get paid at the gym as well when I go. So a passion is what pays you, right? So both science and fitness now both are my career. And why I share this today is just one small action which I took as a 5K runner. I came to a journey where I became an instructor, a journey where I could produce more runners, and I have my own running club. So your journey can be much, much bigger. I got to know all this very recently, but imagine if you can invest that kind of, cultivate passion and invest that kind of a time at this young age, you will excel a lot. So start seeing what is your journey that you want to have for your life. It's one life, live it to the fullest, right? So if you look into the words which were my journey, there was passion, there was focus, planning, actions, progress, upgrading myself, evolving always. So these are the terms which is needed to have a vision. Your core skills are different that you get through your education. But these skills, personal skills, these will take you to a journey, an extraordinary journey. That is what I want you all to have a vision. And I'll not go much into detail, but today I encourage all of you to have that grand vision for yourself. Start looking into what is that you want to do in next 5 to 10 years. If you do not dream today, if you do not think for your vision, you're just going to stay as a normal human being who will just end up finishing masters, going to some job, whether you're interested or not, and then just maybe get married, have a child. I mean, of course, that is probably for most of us, which we do. But when you are passionate about certain things, the energy that you get is also very different. And you will live the life with utter satisfaction. That's what I'm saying. Living a life full of satisfaction, full of energy is different from just living a normal life. And you are all very young. That's why I want to encourage you all. And if you look into your career, any career roadmap for a life science student, bachelor's and master's is what you will take. Either some of them, based on their interest, they move into PhD, or they would go into an industry job. And an academic career, if they think after PhD, they want to move into certain research institutes, become professors, or in certain colleges, if their teaching is what their passion is. So nothing is wrong. It's all about your interest. Choosing the career that you want, that you are interested in, is what makes you. You don't have to be influenced by what others do. So these are the only routes that you have. Of course, the next level is you can become entrepreneurs based on what you, again, based on to become entrepreneurs, you need to have an extraordinary journey, extraordinary energy, because that's a different ball game altogether. So this is the roots. So consider your vision, start building that that vision of yourself 5 to 10 years within this arena, I would say. A lot of us, we only see, this is like a tip of the iceberg. We only see 5 to 20% of the job, which I was also doing that. And the below 80 to 95% is not seen. And we don't even know where it exists. And look at that top 5 to 20%, all of us are actually shooting that. So do you think out of 10,000s, everybody is aiming for that 5 to 20%? And you will get disappointed. You will see that, oh, there's no much opportunity, there's no much job, but everybody has an opportunity in this world. It's just that how you position yourself and how you get it. And yes, so let's see what is that 80 to 95%. One is building that awareness and career choices. Second is networking. Networking is very, very important these days. For me, I think my first industry job was through networking. The more you're connected to the people where you want to be, that's when they're going to take you there. So networking is very important. Is everybody still with me? All good? We will, is it getting late? Okay. So let's finish this very quickly. So career opportunities in biotechnology, or let's say drug discovery, drug development. If you look into this cartoon, we have R&D, we have commercial operations, services, and different operations. So in R&D, you have discovery, research, preclinical, you have product development, clinical, and regulatory affairs. So your core skills is always your life science skills. It could be cell biology, molecular biology, any field that you choose in life science. It's all there that you gain it through your education, that you may gain it through your first few research projects or through few industry jobs, first few jobs. And then you start your next transition in career starts after that. You're accumulating other skills, right, which is relevant to different industry positions. So what I want you all to do is, all of you are on LinkedIn. So what you do is first have a vision, but where you want to be, what job role you want to take, either industry or either academic, it's all your choice. But start having looking into the similar job profiles. What is job description? They would have mentioned these are the eligibility criteria, these are the skill sets needed for the job. So start accumulating. It's your project. This is the project of your career. So it's work on this project for you and start seeing how is that you need to acquire certain skills, soft skills or technical skills. You can do informational interviews with experts, go meet with experts and understand how their job is day to day. If you're picking molecular biologists, ask the molecular biologist how they're doing their job in regular, understand that. So that in different ways, you could do short-term courses, projects, internships, read current articles and trends, networking, and have mentors. Have the right kind of mentors. Your career decisions should not be taken by your aunt, uncle, your relatives. Right? Don't ask, should I do this? I think all of us do that, right? Uncle and aunt, they become big advisors. Do not do that. Ask your experts. Experts in the field that you are going to. Okay, so I'm just ending this. So the job market opportunities for you. If you look into the drug development pipeline, most of the life science people drug discovery and drug development pipeline. So this is your zone of opportunity. So start having the skill sets in these job roles. Identify what are the job roles present on this end of your drug development. Every technology relates to drug discovery or any kind of treatments or therapeutics. So all the job roles are here. So start aligning yourself based on what you want to become and what kind of skill sets that you need to acquire for the job roles that exist here. And of course, if you want to pursue the other side, you could. I think this is an important one. I'll just explain this part. When you take up any decisions for your career, you need to hit the sweet spot. What is that sweet spot? Your skills, you're having the right kind of skills, right kind of interest. If you want to be a molecular biology, you don't want to be in computer science, right? Your interest should align with what you want. Values. Values can be, maybe you want any organization you want to work with, you want them to trust you, you want them to identify your skills, you want them to give you opportunity to grow. So these are certain values, you want them to trust you. So when there is all of these, your skills, your interests and your values match, everybody hits the sweet spot that is called career satisfaction. That if you are in this spot, you will not change your job. If any of these three gets it's imbalance, you leave the job. You'll keep pushing yourself 1 year, 2 years, then at one point you will quit the job. So that's what I wanted to share. We will not go into goal setting. I think smart goal setting everybody is familiar with. I just brought in here if you wanted to know about goal settings. So at our labs, we have also started a career excellence program, but based on some of the interview questions that we had with graduates. So they find that apart from their core skills, they need clarity, right? Probably you don't have clarity where you want to be. Maybe I said have a vision. Maybe you don't know how to have that vision. You don't know how to get that clarity. So career exploration, what opportunities are there, how you should explore them. Your digital presence, personal branding is very important these days. You have to make noise for yourself. Only then you're seen. Because there are pool of talents everywhere. You have to make noise through your digital presence and personal branding. Technical expertise, of course, soft skills, job application is another big process. How to face job interviews and salary negotiations, very important. If you do not negotiate your salary in the first case, you will be in the same salary for next 2 years, 3 years. If you compound it in 10 years, you would have lost some 5 to 10 lakhs. Maybe you could have invested and bought something for yourself, right? Which was just a conversation with your employer. It costed nothing. It was just a conversation and positioning yourself with an employer to negotiate your salary, which can give you that financial help or freedom. So this is what the program is we are doing at our labs, apart from the hands-on in wet lab and all of that. So I have my phone number here and email ID. You can anytime reach out to me. As you said, all of you are in LinkedIn, so you can type this name, Merck Heinz Skill Development Center. You will be able to join our LinkedIn group. That's the last slide for you all. Okay, and this is the upcoming training that we have in mammalian cell culture, which is happening from April 11 to 14. I thought I'll just share if any of you are interested. We're going to teach all the best practices in mammalian cell culture. I will also talk with your principal ma'am today how we can have future collaborations so that you can upskill yourself and get more scientifically competent for your future career transitions. And thank you so much. I did take a lot of time, but thank you so much for listening to me. These are the students who came to our lab. We make sure we follow COVID appropriate protocol. So every day we give them the face shield and all the vest. Everything is given from the company. So we make sure you are fully covered and maintain distance while you work with us in the lab. So thanks so much, and I wish you all the best. Hope all of the insights that you got today will give you a new beginning. Yeah, all the best.
Thank you, ma'am, for your technical talk, introducing us to the universal CAR-T cells using Cas9 technology, and also sharing your journey that got you to this place and inspiring our students to become an all-rounder. Now, on behalf of our college, we would like to honor you and show you our gratitude for your time and your presence. I request our principal ma'am to do the honors. Coming to the end of this program, I would like to heartily thank Sri Narayanappa sir, Managing Trustee of Padma Shri Group of Institutions, Sri Satish sir, and our Principal ma'am, Dr. Anuradha M, for the constant support and guidance. I also heartily thank our guest speaker, Dr. Keerthana, who humbly accepted our invitation and for giving us such a wonderful talk on the same. Thank you, ma'am. I would also like to extend my gratitude to the heads of the department, faculty members, and all our dear students for making this event a success. Now, feedback forms will be sent to you. Class teachers will post it on your group. Please fill it and submit it. Okay. Thank you one and all, and have a good day.